Welcome, everybody, to today's Childhood Cancer Speaker Series lecture with Dr. Carl Koschmann, entitled Therapeutic Targeting and Correlate Tracking of H3K27M-Mutant Diffuse Midline Gliomas. Dr. Koschmann is an associate professor and the Chad Carr Pediatric Brain Tumor Center research professor in the pediatrics department at the University of Michigan. He is also the Chad Carr Pediatric Brain Tumor Center clinical research director and the Children's Brain Tumor Foundation co-scientific director. His lab investigates the molecular mechanisms by which mutations in the pediatric high-grade glioma and diffuse midline glioma promote tumor growth and affect response to therapy. His longstanding NIH-NINDS-funded project on the role of ATRX in pediatric high-grade glioma has uncovered its impact on cell cycle control and targeted therapeutics. Additionally, his lab has established an IUE mouse model that is driven by pediatric high-grade glioma drivers. This model is the basis of translational studies supported by the U.S. Department of Defense Cancer Translational Team Science Award and an NIH-NINDS R01 studying the efficacy and mechanism of ONC201 treatment in DMG. He also serves as a PI and PI for multiple early phase studies in pediatric high-grade gliomas and DMG. His work has been published in many high-impact journals such as Science Translational Medicine, Cell Reports, Neuro-Oncology, Journal of Clinical Investigation, Cancer Discovery, and Clinical Cancer Research. We are also proud to call him an ALSF awardee, receiving a Young Investigator Award and multiple post grants. Now I will hand it over to Dr. Koschmann.
Thank you, Lisa. That was a nice introduction, and excited to be talking for this ALSF community. Yeah, as Lisa mentioned, we'll be covering targeting diffuse midline gliomas and a little bit about pediatric high-grade glioma. Glad that so many of you could join today, and looking forward to a good discussion. Yeah, as Lisa mentioned, I'm an alum of ALSF YI grant, and maybe, sort of one of the goals of this program is to pull researchers such as myself from being a pure clinician to having a research component to their career. And I think when I got this grant, it was not so clear to me that I would run a lab down the road. And I'm glad that ALSF helped push me in this direction. And I'll give a shout out to VJ, who was another alum from that year, and also now a brain tumor translational researcher at Sick Kids. I found this picture on the internet. He's apparently also an Oilers fan. So anyway, obviously I remember that YI summit that I would attend fondly, and continue to feel a part of the ALSF community. So we're going to go through these three sections of a talk, sort of like in thirds. I'll stop between there to see if there's questions or anything in the chat. Otherwise, we'll have some time, hopefully at the end, to answer questions. But really, we're looking at drugs targeting H3K27M, DMG, or PDGFRA, altered high-grade glioma. And then you'll see sort of a trend of, so that if there's a single agent with some evidence of efficacy, how might we target resistance, assuming that for these higher grade heterogeneous tumors that we're going to need more than one drug. So both of these projects have a component of that. And then the third part is, how do we track whether patients are responding to treatment? So we'll dig into that as well. Very quick introduction. Some of you, probably most of you, familiar with this tumor. DMG was defined in 2016 by the WHO as tumors arising from the midline, thalamus, brainstem, and spinal cord that are at least grade 2 gliomas. We know now that about 2/3 of these have this mutation in the histone gene, specifically most often at this 27th lysine, which has very strong impacts on what happens with the DNA wound around the histone and the nucleosomes, large-scale transcriptional changes, keeping these tumor cells more stem-like and more proliferative. We know that even within high-grade glioma, which has a very tough prognosis, having the histone mutation actually carries an even sort of tougher prognosis. But most of the talk today will be focused on this patient population. The first part we're going to study, we're going to discuss ONC201, which many of you be familiar with. We're going to discuss some of our published data, but then also sort of where we're working at with this compound currently. And as you may know, this is a drug that was originally designed to be a dopamine receptor antagonist, so closest in class to an antipsychotics, but we know that DRD2 is upregulated in a lot of cancers, including gliomas. There was an early phase 2 adult in GBM, which was mostly H3 wild type tumors, where the drug was found to be very brain penetrant and very safe, but not very efficacious. But fortunately, there were some younger patients on that trial that had H3K27M that did better than expected, and that led to a phase 1 trial that we took part in that was led by Sharon Gardner at NYU, looking at its impact in K27M tumors particularly, but not necessarily with an understanding of why mechanistically this drug might have an effect in this group. What did help was this paper that came out in 2019, which shows that ONC201 separately enters the cells and activates a mitochondrial protease, this CLIP-P, which then allows it to sort of excessively cleave proteins and reduce the function of the mitochondria. This paper was in leukemia, but our group and others have shown that this is indeed how it is working in midline glioma and may actually be the more important mechanism of action. So we looked at results from that phase 1, that's this ONC014, patients that got the later defined phase 2 and 3 dose, as well as patients from an expanded access protocol, just to see if there was a signal. Obviously, these trials were not designed to look for or confirm efficacy. But what we did see was that in non-recurrent patients who got this drug after initial radiation but before recurrence, certainly an impressive median overall survival compared to historical controls, with progression-free survival ranging from 5 to 9 months from the start of treatment, and somewhere around 12 months from diagnosis, which also compared favorably. In order to be able to figure out if this was a signal or if this was just how well patients with K27M are now being managed. For example, patients are increasingly getting reirradiation. These are trials, so maybe there was a benefit just to being enrolled on a trial. We wanted to compare these data to recent trial-enrolled H3K27M patients, and so we looked at the PNOC-003 data, which was a precision medicine trial for brainstem K27M, and you do see that for the first 40% of patients, they have overall survival or PFS that's not a lot dissimilar from these patients on the PNOC trial, but there is a tail of patients that do significantly better with this ONC201. And with the PFS, and this is in brainstem K27M, the average extension was 3 months. Again, these are retrospective comparisons, but promising that there is a signal for this drug. Just looking at spider plots, waterfall plots, a couple of the trends that I would just draw your attention to is that for some of these non-recurrent patients that do have a response, that is a very sustained response. So just this many patients that are living for this long is novel for this tumor with such a tough prognosis. Most of those longer term patients on ONC014 were non-recurrent, so in the sort of starting post recurrence. If you look at the 018, which was the expanded access, you do start to see more patients in the recurrent setting. And many of those patients, as you see here, just grew. The tumor grew. Being on ONC201, the tumor grew anyway. But you do see a large pack of patients with recurrent tumors that also seem to have a sustained signal. And some of those do end up being pretty significant radiographic responses as well. We've done some work and ongoing work looking at advanced imaging with perfusion and diffusion that we won't go into today. So then we worked in our lab along with my colleague, Dr. Vanetti, who's here at U of M, to figure out, okay, why is this drug potentially helping in these K27M patients? Just looking at metabolomics and RNA-seq data from treated K27M cells, we do see that this drug has an impact on glucose metabolism, either through the TCA cycle or glycolysis. It's effective at knocking that down. Most of the enzymes involved in the TCA cycle are actually downregulated within days of ONC201 treatment. But what you do see is an interesting sort of sustained or even increased amount of alternate sources of energy, glutamine metabolism, leading to alpha-ketoglutarate. This led to an interesting finding, which is an alpha-ketoglutarate derivative, which is L2HG. You may be familiar with D2HG, which is upregulated in IDH mutant tumors. This is the other isomer we've shown through a number of studies that this is consistently upregulated in response to ONC201. And we've even looked at this in spinal fluid and shown that patients being treated that have K27M that seem to be having a response to ONC201 do have an upregulation of alpha-ketoglutarate and this L2HG. And one of the things that was interesting is that L2HG is a known inhibitor of lysine demethylations. And one of the things that we have seen and persistently seen in cell culture across sensitive models is that H3K27 which is a repressive mark that is low in K27M cells, in fact, is a hallmark of these tumors, is increased, upregulated within days of treatment of ONC201. And so this was our first clue that why this drug may be having a particular efficacy in K27M cells. And where I became a believer in this mechanism of action was when we looked at tumors at autopsy from patients who were treated with ONC201. And so just to explain this, over here, this is what a trimethylation mark stain looks like in K27M patients compared to those who don't have the mutation. And what we saw in patients who were treated with ONC201 at autopsy is that the tumor actually stains closer to what an H3 wild type tumor would look like, or a normal brain also has high trimethylation. And that was not the case in autopsy specimens from patients who didn't get ONC201. We saw this at U of M. We did another site at Children's National. They saw the same thing in their studies. And so this was the first evidence in K27M of a drug in patients taking away a pathogenic phenotype. In fact, it's the first evidence of that in a K27M study to date that we are aware of. We also looked at this at the ChIP-seq level, and indeed, the longer you're on ONC201, the higher amount of K27 trimethyl enrichment. So we know the drug is getting into that brain, getting into the tissue, and changing its epigenetic phenotype, and maybe helping to explain why this drug may be working better in this patient population compared to H3 wild type or GBM. So this is sort of how we summarize this. That's a lot of concepts, but we see ONC201 going in, disrupting the TCA cycle, glutamine metabolism going up, 2HG leading to an inhibition of this demethylation and sort of turning these cells that have this epigenetically driven program turn back down. We've since looked to see, is there genetic predictors of response, right? So we saw, some number of these patients and, many clinicians who have used ONC201, there are patients who grow through ONC201. So it is not blanketly curing this tumor. Even in patients who have response, the tumor is ultimately regrowing. So we need to be able to better predict who might respond to ONC201 in the small number of patients, not that small at this point. We're looking at many dozens of patients who have had sequencing at U of M and gotten ONC201. EGFR is the strongest predictor of resistance, and MAP kinase alterations are the strongest predictors of response. This is an example of 3 patients, 3 with H3K27M and EGFR. You see their tumor grows, it becomes metastatic in a couple of these cases, whereas a handful of these MAP kinase altered patients were patients that had these really sustained responses. But then we worked with a couple of people in my lab and Sebastian Wozek at the EPFL as to say, what are the genetic characteristics of patients with H3K27 who do well in general? And one of the things we found was that having a MAP kinase alteration may be actually a lower risk biology in general. And if you see, this is a group of patients where we had survival beyond 3 years across a couple of sites, and you see only a handful of them got ONC201. Some of them did, but many of them got other treatments or no treatments and still had that survival. We looked, and indeed MAP kinase alterations, BRAF, FGFR1 being primarily the biggest predictors, but also NF1 and KRAS and possibly PTPN11 are different, are higher likelihood to be found in long-term survivors. We've looked at a methylation perspective. In fact, these tumors do cluster separate from the other H3K27M tumors. This has since been shown by Paul Castel's group in Paris as well. So on the positive prognostic side, it may be that MAP kinase alterations are not necessarily a specific response to ONC201. But what can we learn from the patients on the progressive side and who didn't respond? Is there something about EGFR that's particularly affecting biology? We've looked at this. You don't have to have an EGFR alteration. The more EGFR you have expression level, the less likely your tumor is to radiographically respond to ONC201. And zooming out to all cancer types across the PRISM cancer cell line atlas, EGFR expression and dependency and EGFR IS and sensitivity are all correlated with resistance to ONC201. And indeed, just having ClpP gene dependency also in that, which is the target of ONC201, anti-correlates with EGFR expression. So Sunny, a student in my lab, has been working on this project. He created models of EGFR alterations added to a K27M cell line, and we have seen that that does increase resistance to ONC201. That's actually compounded if you add hypoxia. And we think that's a sort of a separate resistance mechanism that may not be even working through EGFR. There's a lot of data to tease through here, but what we are seeing is that EGFR altered tumors have less apoptosis with ONC201. As well, they grow better at baseline, but they also grow compared to empty vector more on ONC201. And one of the things that's one of our leads for how this might be going on mechanistically is just bringing you back to this concept that ONC201 restores or increases this trimethylation. So that's what we expect to happen in cells that are sensitive to ONC201. And one of the things that Sunny has seen is that actually EGFR alteration itself added to K27M makes the cells have even less trimethylation. And when you treat them with ONC201, that's further compounded. And so we're not seeing that epigenetic shift with ONC201 with these EGFR alterations. And so there is some disconnect either in how the metabolic state of response to ONC201 in the first place, or just that translation to the epigenetic alteration. And so we're going to tease out this mechanism further. And then the second piece of this is, what do you do with that? And so this is work that's been funded generously by the Yuval Tawari Foundation. How do we target EGFR in midline glioma? We know that on the adult side, GBM tumors have struggled to find EGFR inhibitors that are CNS penetrant and effective. But we also know that the EGFR alterations and just overexpression of EGFR and K27M is a different biology than adult GBM. And so we want to have our own class of drugs that are really validated on K27M plus or minus EGFR models and bring those to the clinic, ultimately in combination with a drug like ONC201, because we know that these drugs are not going to work for long periods of time by themselves. So we have 3 novel EGFR inhibitors that I unfortunately have to call compound A, B, and C because they're through data use agreements, and we are not allowed to speak about the names of the compounds just yet at this point. But I will say that these compounds are showing interesting efficacy, exciting efficacy across different models, and that efficacy is really governed by the alteration that we add in. And so we may need to have multiple EGFR inhibitors for different EGFR alterations. Interestingly, Rob and his team in my lab showed that one of the compounds works really strikingly well, particularly in these exon 20 EGFR alterations, which is really a unique alteration that's seen very highly in midline gliomas. So more to come on this. We'll be moving into orthotopic models and hopefully being able to bring some of these to the clinic through this project funded by the Tawari Foundation. So just finishing up the first section of this, I think just being clear about the clinical experience, these are preliminary studies in phase 1 and expanded access, but they compare favorably to recent clinical trials. in the recurrent setting, the overall response rate that's been published by in JCO showed that a RANO HGG LGG response rate of 30%, which is very impressive. On the front side, in the upfront setting, these need to be looked at in studies that were designed for efficacy. There was a commentary in JCO last week, which calls for these studies to be completed before ONC201 becomes standard of care at the upfront setting. And I couldn't agree more with the need for these studies to be completed on the upfront setting. And to be able to figure out whether ONC201 should be moved into the upfront setting as a standard of care. I don't think we're there yet. I do think we struggle as clinicians because families are sometimes ready to be there, and the drug is not yet FDA approved. So it's a difficult situation to be in for a drug that doesn't have any proven drugs to try to attempt to randomize families to a placebo. So sort of a topic for another discussion that's ongoing. But these studies are ongoing through the ACTION study in BioMed, and then also in combination with other compounds through PNOC22. We talked about how ONC201 works through metabolic and epigenetic disruption of H3K27M, and that the alpha-ketoglutarate increase, which has now been seen across a lot of labs, may be driving the epigenetic changes. We also found that MAP kinase shows improved response to ONC201, but also shows improved response to other therapies. So this is likely driven by underlying biology. I'll show some later data that some of our long-term responders don't have MAP kinase. And so we know that you can have response to ONC201 for long periods of time without a MAP kinase alteration. But this may be something that needs to be increasingly brought into the diagnostics of K27M to sort out the underlying prognosis. EGFR drives ONC201 resistance, and that this reduces ONC201 efficacy, it reduces apoptosis, and that this may be epigenetically driven as well through reduction of H3K27M with methyl-3 with ONC201 treatment. So we're working on a consideration of co-treatments in the future. That was a lot. We're going to move on to the second part of the talk, but I'll stop and see if anybody has questions. The next part will be about targeting PDGFRA, so sort of moving away from ONC201 for a section. All right, maybe I'll just move ahead. So this is a project that we've been ongoing for a little while in collaboration with Mariella Philbin at Dana-Farber and Johannes Goyo, who is in Vienna, and grad students and postdocs in our labs looking at avapritinib in PDGFRA-driven high-grade glioma. As many of you know, PDGFRA is the most common growth factor receptor alteration across high-grade glioma. You really see alterations really across the entire gene. These are activating gain-of-function mutations or indels. We've sort of further characterized the co-occurring mutations, but there are a lot of the common actors in high-grade glioma. Our group had worked historically with dasatinib, which is somewhat CNS penetrant and targets PDGFRA, but it is also relatively dirty tyrosine kinase inhibitor. This is a kinome inhibition tree, and you see that it has activity against a lot of tyrosine kinases. Avapritinib is relatively clean on that, and so that was one thing that led us to be interested in studying this. We've also found that it is blood-brain barrier penetrant. This is kinase inhibition assay, so a little flipped on the head of dose response curves, but what was impressive about it is that avapritinib retains activity against these loop domain D842V mutations, and that's how it's being used in adult solid tumors like GIST. We've worked across our labs and shown that it's really nanomolar efficacy, kind of regardless of the PDGFRA alteration. It's very effective at reducing phospho-PDGFRA, even at really low doses. So that's promising. We've seen that over time, total PDGFRA expression on the surface is reduced across these models. We moved into the mouse model. In our lab, we use this IUE model, intrauterine electroporation, where we inject plasmids prenatally and then can modify what alterations are on those plasmids we adapted this model from Tim Phoenix's lab. And we do see that these tumors grow with the H3K27M and the loss of trimethylation to replicate the human biology. And what we saw is that avapritinib is effective in this model, certainly suppresses growth in early time points. This is an extremely aggressive model, so survival is increased on the order of, sort of 20 to 30 days, but that's actually, very impressive for this model, which is otherwise unradiated and very aggressive. We do see reduction of phospho-PDGFR, very striking reduction across this and another model, which is reassuring. That's seen down here. And then working across our 3 clinical sites, just at our early clinical experience with this drug, we do see some responses, including some really dramatic responses. This case 1 is a patient with a PDGFR amplified tumor. And what we see just within a few months of treatment is that the contrast-enhancing disease is completely reduced. And that persisted for that patient for quite a while. We also see reduction in pontine lesion here and a cortical lesion in case 2. While we were working on this drug, again, we sort of had this question of this drug by itself is not going to be able to eradicate high-grade glioma. It's too heterogeneous, even in our patients who have a response to avapritinib in these early anecdotal experiences. The tumor does regrow. We do know that those tumors at that point often have shifted their genetics. And so Callan started to look in our mouse models of what might be the clues to resistance to this drug. And while PDGFRA goes down, phospho-ERK, which is one of the downstream kinases from PDGFRA, is upregulated. He saw this in cell culture as well across models, that even in short periods of time, the more avapritinib you use, actually, the more phospho-ERK goes up. And so since phospho-ERK is upstream of a bunch of transcriptional proliferative profiles, we thought this might be a marker of resistance. And so we worked with Jean-Philippe Capet from UCSF, who's developed this really cool high throughput kinase activity mapping assay, which allows us to look at hundreds of kinases, the actual kinase activity at a protein level in real time. And indeed, what we see when we treat K27M cells that have a PDGFR amplification with avapritinib is a marked reduction in the activity of PDGFR and KIT, as we would expect, but MAP kinase going up even within 8 hours of treatment. These are all multiple enzymes within the MAP kinase pathway. And even cells that we chronically treat with avapritinib, we see that MAP kinase upregulated. And just starting to look under the hood, when we have access to this kind of kinase data, where might this resistance phenotype be coming from? The kinases are many, and they are certainly interacting with each other. And one of the things we see on the short term is that it is, sort of canonical MEK upregulation, but also MKK4 and 7, which are upstream of junk. But when we look at chronic treatment of cells, it actually starts to really localize to MEK2 and enzymes that are immediately related to that. And if we go back to our short-term, MEK2 is actually the strongest upregulated or strongestly active kinase after treatment with avapritinib. So we've been addressing this with co-treatment with trametinib, which is a MEK inhibitor, and a more recently developed ERK inhibitor, ulixertinib, which do show nice synergy in vitro. We see that they both just showed trametinib data here can knock back down that ERK when co-treated with avapritinib. And there's some very preliminary mouse data. This is actually flank data that avapritinib is certainly effective at suppressing the tumor, but starts to grow faster than these co-treated tumors with avapritinib and trametinib or ulixertinib. So we're going to be moving this into mouse brain tumor models. We actually recently used a tumor organoid drug screen on a patient who had a PDGFR-driven sarcoma that was resected. This was a CNS met of a rhabdo that was PDGFR mutant. And this tumor had grown on avapritinib. So when we took the tumor and grew it using this organoid protocol that we developed, basically adopted from the Children's Brain Tumor Network, we were able to see that the tumor is indeed fairly resistant to avapritinib. The patient had been chronically treated with avapritinib, but that retained sensitivity to trametinib and some synergy there. So this patient has since been treated on avapritinib and trametinib for the last 5 months with stability. Again, sort of a N of 1 here, but maybe some feasibility that this might be a future therapeutic angle. So just summarizing this second section, we see PDGFR alterations are common in high-grade glioma. Avapritinib is selective for them, is effective, has good blood-brain barrier penetration. I didn't show that data today. And then some early clinical activity. In terms of how might we think about combinatorial therapies, we are seeing pretty consistent upregulation of MAP kinase activity that's really driven by MEK. We've seen it now at the single cell level that's actually occurring primarily in cycling cells. We didn't go into that data today. And then again, combinatorial therapy, some early evidence that this might build off the success of avapritinib. So I'll stop there. Otherwise, I'm ready to cruise through to the final section on correlate markers. And then maybe handle questions at the end unless anybody has any now. I see there is a question on ONC201 I'll maybe circle back to at the end. So how might we track response to ONC201 or avapritinib or other promising drugs in midline glioma? We know that MRIs are certainly the gold standard, but often lead to areas of frustration, not knowing if tumor is growing or you're seeing radiation effect, or just the ability to potentially have another metric of response. We had worked with Amy Bruzek some years back to collect spinal fluid from patients being chronically treated with ONC201. And here's a patient who actually had P53, PI3 kinase, and H3 mutations and had a sustained multi-year response to ONC201. And we were able to track that her spinal fluid amounts of these tDNA did increase. They actually increased initially and then decreased over time. And then she had a sustained response to ONC201. We worked with Evan Cantor in my lab when he was in my lab to collect CSF from patients on one of the arms of the phase 1. These were patients treated after initial radiation, and we collected CSF at 0, 2, and 6 months. We've since gone on to additionally collect CSF from these PNOC studies to be able to see how can we, on a study-wide perspective, look for how tDNA can help understand whether patients are going to respond or in the process of responding or growing. This is a lot of data, and I will just summarize it by saying we looked at this in the CSF, this is all blue, and in the plasma, this is all orange. This Evan's work was with a ddPCR assay for specifically the histone mutation in these biofluids, and we do find that when we use some methods to increase the signal, such as like pre-amplifying the H3 loci and then vacuum centrifuging and then doing a ddPCR amplification on that H3K27M, you can find this in the majority, certainly almost all of the CSF samples, and even above 90% of our plasma samples if we get a reliable signal for H3. Tracking those over time, the CSF is a strong changes in allele fraction, the amount of mutant versus wild type are a stronger predictor of response in CSF compared to plasma, but you also see a trend that reduction in plasma H3K27M can predict longer response to ONC201. Here's an example of a patient who had a sustained response to ONC201. Her tumor initially grew radiographically, that's this gray line here, and actually almost grew enough to get her pulled off the trial, but we kept her on the trial, and you see that whole time her plasma tDNA is dropping. So we didn't have this data till later, but you might imagine as a clinician, if you had these extra data points, you might feel more reassured sticking with the treatment and attributing this to radiation effect, which it certainly was in this case. Here's a patient whose well, we'll, in the interest of time, move on to this next case. Here are examples of how spikes in these allele fractions, when what we considered a spike was a 25% increase in the allele fraction from that patient's baseline. Those can precede progression. So here's an example of a patient whose tumor was shrinking on ONC201, but then ultimately grew. What we saw in both plasma and CSF was that allele fraction was increasing even as radiographically the tumor was shrinking. So again, maybe obviously we're not going to replace MRI, but maybe these pieces of information, if they could be performed in a CLIA setting in real time, could potentially help clinician and family understand what's going on with the tumor. And then just going a little further with this, Jack Wadden is a postdoc in my lab who's been working on other markers. We can sequence tumor DNA in spinal fluid. We can use digital droplet in CSF. We can use digital droplet in plasma, but can you sequence in plasma? Can you find enough tDNA to be able to look across multiple alterations in plasma? This has yet to be done in brain tumors. And one of the things that Jack has tapped into is using what we call concatameric sequencing, which is actually been a concept that's been rapidly developed for some COVID diagnostics to try to be able to improve the detection and confidence of detection of tDNA, because we know in plasma tDNA it's there, but it is exceedingly rare. So we've got few events and we need to capture and sequence them. Just one case that where he sequenced for H3K27M and BRAF in the plasma. This patient, there's a lot going on here, but this is the tumor area in black. She was treated with ONC201 and a BRAF inhibitor over this period of time, actually BRAF inhibitor and a MEK inhibitor. And what you see over that time is that the K27M is increasing. And that is occurring both by digital droplet, but also by sequencing. The ONT is our nanopore sequencer that we're seeing. But then sequencing the BRAF, this actually goes down. And so this separation here, we weren't sure if this was a real separation or if this was a failure of our sequencing instrument. It's not common for us to see in patient tumors that a driver goes away at autopsy. And it's relatively new that we're treating these BRAF mutant tumors with BRAF inhibitors. Well, so what we looked at then was CSF at this point. We worked with the Memorial Sloan Kettering team, and they did CSF sequencing at this time point, and indeed the BRAF mutation that was found at baseline was no longer there, that the drugs had actually selected against that clone. And if you look back to the initial sequencing, this patient did have clonal H3, but it actually may be slightly subclonal BRAF. So some section of this tumor was BRAF negative, maybe a third of the tumor. And that ultimately, we think, grew while the patient was being treated with the concurrent BRAF inhibitors and took off. We also were able to find a fusion on that CSF that may have also been part of the actual escape and growth here. So another example of how we may be increasingly able to move CSF, certainly helpful. Plasma may also be helpful clinically to be able to look at multiple alterations. Little further into this, so this is a PNOC trial that I work along with Sabine Mueller and Javad Nazarian to co-lead. And what we see across institutions is the ability to treat ONC201 the ability to treat DMG in multiple different time points. So upfront, you can be put on treatment after radiation, and then again at progression. And this is a platform trial. So in its first cohort, we used ONC201 with the PI3 kinase Paxilicib, based on Matt Dunn's data that these two drugs work well together. But that trial is built actually to set up for multiple ongoing and future platforms of combinations. And what we did on this was to employ the same sort of like progressive plan of collecting lots of samples, including CSF and plasma. And I show this data to show that we have gone back to that CSF metabolomics that I showed earlier on ONC201. These patients now treated with ONC and Pax. And again, we're seeing that L2HG and glutamate changes are some of the strongest predictors of overall survival on this trial. So, the systematic use of CSF and the ability to look at not only tDNA, but also things like metabolism may be useful in looking at response. And then finally, can we move out of the liquid into MRI and MR spec? If you are familiar with adult IDH mutant tumors, they are now looking for things like 2HG with MR spec as a response to IDH inhibitors. And what we've seen working with Pavitra Viswanath is that we do see glutamine metabolites that are affected by ONC206. In particular, what Pavitra has seen is that GABA, which is a derivative of glutamine, is upregulated in cells and mouse tumors treated with ONC206. So we're now going to be employing MR spectroscopy in our clinical trials to be able to look for changes in these metabolites that may be able to tell us early on whether that patient may be a longer-term responder. So just to finish up this, from our correlate test, the H3K27M ddPCR is the closest to the clinic. We're getting pretty close to CLIA certification here. From an exploratory perspective, sequencing of CSF and plasma using nanopore sequencing. We didn't talk about this today, but we have an exciting collaboration with the Shima lab to look at H3K27M nucleosomes and TP53 mutant molecules in plasma or CSF. And then metabolite analysis of CSF and MR spectroscopy, again, sort of in that research grade, but hopefully being able to help guide future patients being treated with these studies and having not just MRI, that you could also have MR spec and you could also have liquid assessments to be able to give a more cohesive view on how the patient's tumor is responding. I'll stop there. We certainly appreciate all of the hard work of the people in my lab, our collaborators within the U of M and outside, our funding sources, and then, of course, the families who have participated in this research or donated samples so that we could have the data that we have. I'll stop there. Looks like we've got a couple questions queuing in that I can maybe start to address. Let's see here. First question I have is from Bin Chen. How about the PK profile of ONC201? Some sources show it is not soluble, but dordaviprone is reported to be water-soluble, orally bioavailable inhibitor. So we published in the supplement of that cancer discovery paper that the drug ONC201 deposits in the brain at micromolar level that with sort of like human equivalent doses. We've seen there was a paper published from one of the early GBM studies where they did target validation and also showed a range in drug penetration, but micromolar seen in some of the patients in tumor specimens. And this is a drug that is taken orally, exclusively orally. So I think CNS penetration, this is a rare compound where CNS penetration is not the issue. We've had some families and patients ask about, maybe you could tie this together with focused ultrasound. And I'm not sure this would be the compound I would push in that direction because I think CNS penetration is good. This could be possibly enhanced by using day 1, day 2 dosing, which is being used on a number of these trials. Some data that the second day, the peak in ONC201 is even higher, at least in the plasma. We haven't, I don't think we've looked yet in brains. And then again, there's this whole second wave, this ONC206, which is a modified version of 201, which appears to be even more brain penetrant. Ian asked, what is the true 1-year PFS and OS for newly diagnosed true DIPG with ONC201? I'm not sure what true DIPG means. Does it significantly exceed the historical range of 1 year, 14 months given the range? I think maybe what you mean by true DIPG is true H3K27M. That's at least how we could make some consistency with current and historical treated patients. That's what we've attempted to do. And so if we go back to the to our data, which is let's see, where's that? Way at the beginning. This is two different trials. And so if you're looking for PFS for H3K27M brainstem, that's down here. What we saw in those 20 patients across both those protocols who got the phase 2 dose was a PFS of about 11 months, whereas the PFS in this other trial was 8 months. This put an overall survival of 21 months in brainstem, but this was certainly affected by this little tail of patients that happened to be straight above the midline. These are still small n. These are still two protocols added together retrospectively. So I wouldn't put these as gold standard. I think the current phase 2, phase 3 randomized studies with upfront ONC201 will be the best place to ask and answer these questions. Jeffrey Allen, any evidence that new mutations arising in DIPG are contributing to resistance? Great question. What are the intrinsic genetic predictors of leptomeningeal metastases? Also a great question. So, if you look at our studies and also, maybe I'll stop sharing. If you look at our studies and other studies where we've done autopsies of patients with H3K27M and compared them to their baseline, the drivers stay remarkably similar. Now, a lot of that is from studies where we were using ineffective drugs. So it's possible that in the new era of some of these trials related to ONC201, some of these trials with CAR-T, that we may start to see more genetic drift with treatment. But a lot of the key drivers that show up on your baseline sequencing also show up at autopsy. This patient I showed that lost their BRAF and picked up another fusion, that's a single case of that. We've had other patients here. I'll give an example of a patient who had H3K27M EGFR alteration in a thalamic tumor. We treated with an EGFR inhibitor, and the patient developed a brainstem met, and that had an NTRK fusion in it. We did a resection of that. And so, I think it's an argument for, as we're getting more drugs that are getting into the brain that might be showing a signal, we're going to want to not just assume that the genetics from initial biopsy are stagnant. And CSF sequencing, I think, is a nice agnostic way to look without having to rebiopsy. Increasingly, that's CLIA certified through Memorial Sloan Kettering, University of Washington. Some, hopefully industry will get there as well. So that you can have these checkpoints, when you're at a decision tree, you have a new MET, you have new growth. Let's just recheck what's going on genetically. Rebiopsy, re-resection also to be considered, but obviously, these are areas where, we don't want to take those decisions lightly. I don't think we have great predictors of leptomeningeal metastases. Maybe there are others who have looked at this a little bit further. I do think that when we look at, I'll just go, I think I stopped sharing my screen, but maybe I can go back to sharing my screen. When we looked at patients that had short-term survival, and this is not exactly what you're asking, but what are the genetics of patients that are less likely to push past average historical survival? Those we saw, P53 was the strongest predictor. And maybe that's not surprising. I think many clinicians have seen that clinically, that these tumors are a little bit less likely to respond to radiation. I think what hasn't been done is a good study to look at midline gliomas that develop leptomeningeal disease. What were the baseline genetics that predispose to that? There may not be any DNA level events, but that would be a good study to run. And then also, are there alterations that are being picked up at the time of leptomeningeal disease? So far, I haven't seen any evidence of that, but I think that's a study that needs to be done more systematically. Okay, I'll go back to my chat. See if I think that's my last question from the group. Feel free to add them in. I'm not sure if other people have the ability to talk, but I feel like I've been talking for quite a while.
Hi, Carl. It's Liz Scott. Thank you.
Hey, hi. Good to see you.
You too. Great presentation. I think we do have time if anybody has any other questions. I don't think you can see anybody except the Alex's people because it's
Okay.
a webinar. But people can feel free to ask additional questions if they have them. It was a good turnout today.
Yeah, I'm glad to see so many people interested in this space, and you know, I think it maybe is indicative of you know, we do think we're really starting to turn a corner with this tumor, and with the tools we have, hopefully we'll get even further to help out these patients.